So in terms of energy source, we look at whether organisms can make their own food or whether they have to consume food. And so autotrophs are self-feeders. They can make their own food, and we have bacteria that are autotrophic, but we also have bacteria that are heterotrophic. Can you hear my cat? He's getting old. So the heterotroph bacteria, hetero meaning other, and they have to consume food, and we'll look at types of both of these kinds of bacteria. So I'm going to look at the autotrophs first, and some of us covered photosynthesis in 1408 in the spring. Some of us didn't, but you've probably heard of photosynthesis in your education up to this point. So organisms that use sunlight as an energy source are autotrophic, and specifically sunlight, we consider them photoautotrophs. Okay, so they use sunlight as their energy source. And so we are going to mention some of these cyanobacteria in lab, but cyanobacteria is the general name for those bacteria that are photoautotrophic. They're going to be green. They're going to be able to do photosynthesis. So the ones in this picture here are oscillatorium, which we're going to look at in lab. And then the one here, we've got oscillatoria labeled. We also have anabina labeled. It's the one that looks like kind of a chain of pearls. Both of these are photosynthetic. Both of them are cyanobacteria. Some people call them blue-green bacteria. And they're considered colonial, meaning that there's quite a few cells that are linked together in some sort of a chain. Now, just to mention, we're not going to look at any of these in lab, but there are those autotrophs that are considered chemoautotrophic, and that just means that they're getting their energy source to make their own food from chemicals rather than from the sun. And so a lot of times those chemicals are some type of a mineral. Iron is a big one, and so it releases electrons that contain energy, and that's how these organisms make their food. So in terms of bacteria that are way down deep in the deep sea vents, they don't have access to sunlight, but they have access to chemicals that they can do chemoautotrophic with. So if we talk about the heterotrophs that cannot make their own foods, they have to get food from outside sources. Many of them, in terms of bacteria, are decomposers. So that's a general term. Decomposers break down waste. You drive by any of the garbage dumps in this area, and they don't smell very good, especially on hot summer days, and that's because there's a lot of decomposer bacteria in there breaking down things, and as a result of breakdown, they produce gases, and a lot of these gases don't smell very good. So saprotroph is a specific type of decomposer, and they're getting their nutrition through external digestion, meaning that they produce enzymes that are released to the outside of the body. Digestion happens in the environment, and then the organism absorbs that food. So we are not saprotrophs because we have to ingest our food, and digestion happens on the inside of our body. But many bacteria are saprotrophic, meaning that they digest food on the outside of the body. Okay, the characteristic of oxygen, aerobic versus anaerobic. The aerobic organisms would require oxygen. Anaerobe, an means without, and so these would be the organisms that do not require oxygen. So I've put a picture from your book down at the bottom of this slide, and the first picture is of a little mouse, and if he has bacteria living in his gut, in his digestive tract, inside there is anaerobic. We have it also. So those bacteria that are working on breakdown in your gut are usually anaerobic. They don't require oxygen. If you have bacteria living on your body surface, on your skin or so forth, they're exposed to oxygen, they're probably going to be aerobic. Okay, if you are a plant person and you've got plants in pots at your house, if you have a well-draining pot, then there's going to be oxygen down there, okay, and your plants are going to do pretty good. If you don't have a well-draining pot and there's lots of water, that's a good place for anaerobic bacteria to grow, and you may have a plant that's not doing so well. Same thing if we look at a lake. The surface of the lake up at the top is going to be exposed to oxygen, and that's where you're going to find aerobic bacteria. But down in the bottom sediments, there is no oxygen, and that's where you would find anaerobic bacteria. Okay, in terms of reproduction, how do these organisms reproduce and make a new generation? The process that we see in bacteria is called binary fission. So binary means two, and fission means to split. And so you can see in the little animation at the bottom here that you would start out with a single cell. So I'm going to wait for it to go back to the single cell. Has one piece of DNA. That cell is going to copy its DNA, so there's two pieces. It's going to separate the two pieces into two different sides and split in half, and that is binary fission. It's a very simple process. Okay, so the circular chromosome replicates, the cell gets a little bit larger, separates the membrane and the wall, and you have two identical cells. So another term for this process is called vertical gene transfer, meaning that those genes are passing from the parents to the children or the next generation, the offspring. Okay? So this is not a mitotic process. We've looked at mitosis that you see in eukaryotic cells where they make copies of all their DNA and they go through prophase, metaphase, anaphase, telophase, and so forth. That does not happen in bacteria because they only have one chromosome. So there's no need to be quite as organized as we are in mitosis. So you do have a picture in your book illustrating this, kind of like that animation that I showed you. So in bacteria, we have other ways of getting new DNA. And so these three other ways are called horizontal gene transfer. So horizontal means that the DNA is coming in from the side somewhere. It's not coming in from a parent, but they're picking it up during their lifetime. Okay, so I'm going to look at the first one. I have a picture here of the first one. It's called transformation. And this is when bacteria basically are able to incorporate DNA from the environment, actually bring it into their cells. So if a bacteria in the environment dies, it starts to break down, DNA then is released into the environment, and bacterial cells are able to pick up some of that extra DNA and incorporate it. And so then they're going to have new characteristics because they're going to have new DNA and new genes. We cannot do transformation. We can't pick up DNA from the environment. We only get our DNA from our parents. Okay? So just a quickie animation of transformation. So you have this bacterial cell that dies. DNA is released into the environment. You have a new bacterium who has some kind of a binding protein that will allow to bring DNA into the cell. It's going to pick up that DNA from the environment, insert it into its own DNA, and then you've got new DNA in this cell, and it will be transformed because it will be able to make new proteins with those new genes. Bacterial transduction, the second type, is where a bacteriophage, which is a virus that attacks bacteria, brings DNA from one bacterial cell to another. So on the left-hand side of this illustration, you see they've labeled it the transducing phage, but that is a virus, and viruses have DNA in them. So viruses can infect bacteria. You notice that that virus is injecting DNA into the virus in the second figure over. And then the bacterial cell actually incorporates that DNA, and it's been transduced. So you can see here that the bacteria is not just picking up DNA from the environment, but it's picking up DNA from a virus that is carrying that DNA. So I'm going to show you just a quickie animation of this process. So the big thing that looks like a Mars landing pod is the virus, and it has DNA in it, And when it attacks a bacterial cell, it inserts its viral DNA into the bacterium. Okay? And once that happens, it takes over the mechanisms in that bacterial cell, makes new viruses, kills the bacteria, releases these viruses into the environment. And you notice that one of those viral cells or viral organisms has picked up new DNA, which it's now going to insert into the next bacterial cell. And now this bacteria has picked up DNA from a virus. Okay. And then the last one we look at is called conjugation. Now, it's sometimes referred to as sexual because two bacteria are sharing DNA. There's not really such a thing as a male or a female bacterial cell, okay? But we can describe them as plus or minus if they do or don't have this extra DNA. But you notice they form a conjugation tube between the two, and they share DNA. And so when they are separated, now we're going to have cells that have new DNA that they picked up from another. It's still horizontal. They're not picking it up from parents, but they're picking it up from another bacterial cell in the environment. Okay, so here you have the bacterial cell on the left that has some extra DNA, the conjugation tube. And so when these two bacteria exchange genetic information, bacterial cell on the right picks up new DNA, which is going to give it new characteristics because it's going to have that extra plasmid, which is extra DNA. So you do have in your book the 3 types of horizontal gene transfer illustrated, which you can go back and look at if you want to from your textbook. Okay, and then we also look at where bacteria live in terms of a way to classify them. Okay, so we really have 2 choices. Bacteria can either be free living, so they're just floating around in the air and on surfaces and in the soil and so forth, or you can find them in symbiotic relationships, meaning that they're living with another organism. So in this particular example, because I think it's important to have a little bit of an idea of some of the ways that bacteria help us, the nodules are like little bumps on the roots of plants. And so plants that you know make fruit or make root parts that have a lot of protein. So beans we know have protein, peas have protein, peanuts have protein. These are legumes. And they need a lot of nitrogen to make protein. And it's very difficult to get nitrogen out of the air for a plant. So if it has a little bacteria cell living in it that's able to fix that nitrogen or make that nitrogen available, it's going to be easier for these plants to make products that have protein in them. So in this case, the 2 are living together. The plant is basically a protection for the bacteria, and the bacteria helps the plant use nitrogen. So you have a picture in your book illustrating places where bacteria would live on you, and most of these are living symbiotically with you and not causing too many problems. Okay, so we have bacteria in our mouths, we have bacteria on our skin, we have bacteria living in our stomach and our intestines. You got bacteria living under your arms and your nose and your throat. So you have a bacteria living all over, and most of them are not causing a problem. But if we have big overgrowth, then we do have some problems. You know, if you get acne or something, you've probably got overgrowth of these bacteria that are causing inflammation and that kind of thing. But realize that you as an organism are living with a lot of bacteria as part of your body, and most of those bacteria are either not causing a problem or are actually helping.